Hello everyone, this is MMBS-111, Introductory Microbiology. This mini lecture is over section 10.1, Principles of Antimicrobial Therapy. There are 5 learning outcomes for this mini lecture. First, state the main goal of antimicrobial treatment. Second, identify the sources for the most commonly used antimicrobials. Third, describe 2 methods for testing antimicrobial susceptibility. 4, define therapeutic index and identify whether a high or a low index is preferable. And 5, define selective toxicity. So we're looking at antimicrobials in this chapter and how to control microorganisms. Antibiotics were first introduced in about the 30s when Alexander Fleming discovered that a particular mold from the group Penicillium was able to prevent the growth of certain microbes on an agar plate. And since then, we have isolated these metabolic products from a number of bacteria and fungi. And these antimicrobials are natural products that are produced by these organisms to inhibit the growth of other competing organisms that grow in the same habitat. The 4 groups listed there, Streptomyces, Bacillus, Penicillium, and Cephalosporium, are all groups of either bacteria or fungi that we have derived or isolated antimicrobial products from that we currently use today. The main goal of antimicrobial therapy is to administer a drug to an infected person that destroys the agent of infection but does not harm the host cells. And that's really important. Some antimicrobials are better at addressing that than others. There are a number of characteristics that we look for when identifying or trying to either synthesize or isolate an ideal antimicrobial drug. First is selective toxicity. We want the antimicrobial drug to kill the microbe but not be toxic to host cells. So there are certain parts of microorganisms that we try to target with antimicrobial drugs because that helps the drug be more selective to that particular organism and not harm the cells of the host. We want our antimicrobial drugs to be microbicidal rather than microbistatic. Microbicidal drugs kill all organisms that are sensitive to the drug. In a microbistatic antimicrobial, we halt growth, so the organisms are not able to reproduce. So we stop the population from increasing. The problem with a microbistatic is that once the treatment is removed, then the organisms are able to reproduce again. We want an antimicrobial drug that is relatively soluble, so we want these drugs generally to be soluble in water or some other safe solution, and we want it to be able to function effectively even when highly diluted within body fluids. A good antimicrobial should remain potent long enough to perform its action within the body and not be broken down rapidly or excreted by the body prematurely before it's able to perform its action successfully. Ideally, we want drugs that will not lead to the development of antimicrobial resistance. And it should complement or assist the activities of the host defenses. So a good antimicrobial is going to assist the host's own immune response and other defenses to address that infection. We want it to remain active in tissues and body fluids, and it should readily be readily and easily delivered to the site of infection. So whether it's internal or external, that antimicrobial should be able to easily be delivered to wherever the site of infection is. Cost is also an issue. We want this antimicrobial drug to be reasonably priced, and it should not disrupt the host's health by causing allergic reactions or predisposing the host to other infections. When we talk about chemotherapy, there are a number of terms that we need to be familiar with when we're talking about chemicals that we're using as drugs to treat certain conditions. So a chemotherapeutic drug is any chemical that is used in the treatment, relief, or prophylaxis of a disease. And prophylaxis is the use of a drug to prevent infection, generally used to treat individuals who are high risk of developing infections, those who are immune compromised or might have particular infections that make them more susceptible to additional infections. Antimicrobial chemotherapy is the use of the chemotherapeutic drugs to control infections. So we're talking specifically about microorganisms. Antimicrobials is an all-inclusive term for any antimicrobial drug, regardless of its origin. So when we say antimicrobial, it could be a naturally produced compound from a bacterium or a fungi, or it could be a drug that was chemically synthesized within a laboratory. When we use the term antibiotics, we are specifically talking about products that are produced as a natural metabolic process by other microorganisms that can inhibit or destroy other microbes. So an antibiotic is a natural substance. Semi-synthetic drugs are drugs that are chemically modified in the laboratory after being isolated from a natural source. So we might isolate a particular metabolic compound that is produced by a bacterium, and then we might, in the lab, alter it somewhat to improve its effectiveness or to address maybe a resistance issue. If we alter it chemically enough, it can be used again against organisms that have developed a resistance to the natural form. Synthetic drugs are drugs that are produced entirely by chemical reactions. So these are manufactured and produced within the laboratory. They are not found naturally. And then we have narrow spectrum and broad spectrum. So a narrow spectrum chemotherapeutic is an antimicrobial that is effective against a very limited array of microbial types. So it might be effective only against gram-positive bacteria, or it might be effective against a very specific group of bacterium like Staphylococcus. A broad spectrum antimicrobial is a chemotherapeutic that is effective against a very wide variety of microbial types. So it could be effective against both gram-positive and gram-negative bacteria. It could be effective against both bacteria and fungi. So it has a very wide range of effectiveness. So before we start an antimicrobial treatment, there are some things that we want to know first, and that is the nature of the microorganism. So what is the organism that is causing the infection? Is it gram-positive, gram-negative? Is it E. coli, Staphylococcus? Is it fungus? We also want to know how sensitive or susceptible that organism that's causing the infection is to a variety of drugs that can be used against it. And then we also need to keep in mind and be aware of the overall condition of the patient that we are treating. Um, Identification of the infectious agent should begin as soon as possible. And this is really important, especially in the environment now with um, the increase in antibiotic resistance. Um, it really should happen before any drugs are given. This, unfortunately, isn't always the case, especially if you are seeing your family practice physician. Typically, your physician is going to do their best to look at your symptoms, do a rapid test to try to identify whether a particular organism is present, and then prescribe an antimicrobial. Unfortunately, a lot of organisms have similar symptoms. So frequently, antimicrobials are prescribed before the physician is certain of the organism that is causing the infection and its sensitivity to certain drugs. Based on the CDC's recommendations and a report that they issued in 2013, there are a number of organisms that are highly resistant, and treatment of these organisms should not occur prior to identification of the organism and its sensitivity to particular drugs. And those are all of the Staphylococcus species, especially Staphylococcus aureus, Neisseria gonorrhea, Streptococcus faecalis, and most aerobic gram-negative intestinal bacilli. So these are organisms that really need to be identified and have some antibiotic susceptibility tests performed prior to treatment. There are a few tests that are available to assess drug susceptibility in organisms, and we're going to look at these independently. One test is the Kirby-Bauer technique, and this is where you take a large agar plate and you spread bacteria all over the plate, and you create a lawn of growth. We are not trying to streak for isolation. We are covering the entire plate with the organism. And once the entire plate has been covered, then we're going to place small paper filter discs around the plate. And each disc has been soaked in a particular concentration of a very specific antibiotic. And then we're going to incubate that plate. And after about 24 hours to 48 hours, depending on the organism, of incubation, we're going to look at the zone of inhibition. And this is a ring of no growth around each antibiotic disc. We're going to measure that zone of inhibition and determine whether the organism is susceptible or resistant to that particular antibiotic. And there is a standard available to compare the size of the zone of inhibition to determine resistance or sensitivity. And these are helpful in creating what is called an antibiogram. Antibiograms are a set of data that's typically collected multiple times over several years to identify isolates, particular strains of organisms that are present in that location, that locality. So in that particular hospital has seen in that area, and their resistance and sensitivity patterns. This really helps physicians in treating particular organisms. If you know that in Nampa, Idaho, there are these specific strains of organisms that have been seen regularly over the past several months or years. And these are the sensitivity patterns that have been expressed by these organisms. That helps physicians then determine what would be the best chemotherapeutic for that particular organism, knowing sort of the patterns that have been expressed in that location. The Kirby-Bauer technique is not as effective for anaerobic, fastidious, or slow-growing bacteria. I'll let you think about why for a few seconds. So anaerobic organisms are difficult to grow because they have to be grown in an environment that is free of oxygen. So there are additional steps that have to be taken in order to create that kind of environment. Fastidious organisms are organisms that historically are difficult to grow in the laboratory because of their nutritional requirements. So using the Kirby-Bauer technique is difficult for those organisms. And then slow-growing bacteria, we want to read these tests within 24 hours, 48 hours at the latest. And if we have an organism that is really slow growing, it can take additional time to truly assess the sensitivity of these organisms to these antibiotics because they grow so slowly. Another way for us to test drug susceptibility is called the tube dilution test. And most of these tests now are not performed in separate test tubes, but they're actually performed in a micro well plate. And each well of the plate has a different dilution of antimicrobial within the plate. And then each tube is inoculated with a small uniform sample of pure culture of that organism. And then those plates or those tubes are incubated for 24 hours. And then we're going to evaluate growth within those tubes or those wells within the plate to determine where growth has been prevented, what dilution of that antimicrobial has stopped growth and killed the organism. And it's really helpful in determining an effective dosage for particular organisms. So this slide shows an example of a Kirby-Bauer test. You can see that there are different zones of inhibition for each antimicrobial on the discs. You're going to measure that zone and then use your standard to determine whether the organism is resistant or sensitive. It's really important to look at growth within the well because, as you can see in this top space, we have some organisms that are growing here. And then in this bottom one, we also have some organisms growing here. Any growth, even if it's one colony, two colonies within that zone of inhibition, shows resistance. You have resistant colonies here, and you don't want to use this antimicrobial. This is an alternative to the Kirby-Bauer test. This is called the E-test. And the E-test takes strips, and each strip has a different dilution of that same antimicrobial. And you're going to read the strip to the point where the minimum inhibitory concentration is found. So the lowest dilution that will prevent microbial growth. And if you look at this one, we have no growth all the way down to here. So our minimum inhibitory concentration is 0.25 micrograms per milliliter of this particular antimicrobial. Okay. This is an example of a tube dilution test. This is the standard now. Most labs don't use test tubes, but they use a micro well plate. And you can see we're looking for growth. Each little pellet here is growth of a microorganism. And each row has a different antimicrobial diluted at various levels. So this is low concentration of the antimicrobial. This is high concentration of the antimicrobial. You can see here clindamycin, there is growth in every single well, which means that the minimum inhibitory concentration is greater than the dilution in this well, which is 32 micrograms per mil. So we're looking for wells where there's no growth, okay? And that shows us the lowest dilution that will kill all of the organisms, okay? All right, so when we're looking at minimum inhibitory concentration and determining a therapeutic index, what's going to be the best concentration of that drug, we have to consider some other factors, and that is that organisms do not always perform the same in the laboratory or in vitro as they do in the body or in vivo. So there are some things that can contribute to the failure of an antimicrobial that is given to a patient as therapy that has shown to be effective in the lab. And the first is that the antimicrobial may not be able to diffuse into certain compartments of the body like we need it to. Another factor is that there may be resistant microbes present in the infection that we missed when we took the initial sample to test susceptibility in the lab. And then finally, many infections are caused by a mixed flora. We may have multiple pathogens, some of which are resistant to the drug while others are not. So when we're looking at therapeutic index and the best drug or the most effective drug for the therapy and the concentration that's going to be most effective, we have to look at the toxicity of the drug and the dose of the drug that is toxic to humans and that minimum inhibitory concentration. What is the minimum dosage that will be effective against that organism? We want a ratio that is high, a large ratio. We want a significant difference between the toxic dose and the effective dose. So the smaller the ratio, the greater there is potential for drug reactions, for patients to experience adverse side effects. So if you're looking on this slide here, a therapeutic index of 1.1 is risky. There is almost an equal concentration of toxic drug to the effective dose. Okay, so if the toxic dose is 10 micrograms per mil and the effective dose is 10 micrograms per mil, my ratio is 1, and that's not good for the patient. I want a ratio that is large. So if the toxic dose is 10 micrograms per mil, but the effective dose is 1 microgram per mil, my ratio is 10. And that is good for my patient. It means that I can give a small dose that's going to be effective, but it's not going to be toxic to my patient. And that's the end of section 10.1. These are our checking in questions. So make sure you go through these questions and that you can respond to them effectively and correctly. And also make sure you go back and look at those learning objectives that you're able to know, or you do know, and you're able to do those things. Here's our critical thinking question. So in evaluating a treatment plan, the therapeutic index is calculated as a 1. Based on this result, should the medication be used? Why or why not? So think about what we just discussed. Should this medication be used? And be able to explain your answer. The answer to this question is on the next slide, so I would encourage you to come back to the presentation and review your answer. And I hope this video was helpful.